Hello and welcome to another episode of the Omics cast. My name is Albert Vilella. You can follow me on my Twitter account, twitter.com/Albert Vilella, and you can also ask me questions, contact me on LinkedIn. I'll be happy to answer any questions you have. And a reminder that this slide deck is available upon request. Today, we're going to talk about recent advancements in compute acceleration in bioinformatics. Most of what we're going to talk about is genomics, but in bioinformatics in general. And this has been stimulated by this tweet from Andy Carroll, who works at Google, stating that there's a new version of a tool called Deep Variant version 0.7 that uses certain instruction from Intel CPUs that accelerates this process. And then Li Hang, we're going to present him formally in a slide further on, engaged in an interesting thread with Andrew Carroll on Twitter to compare notes on what acceleration levels are provided by these new Intel CPUs. And so bioinformatics has traditionally been a field where specific hardware and instructions have been used to accelerate the compute of specialized tasks. And Google, in their Google Health, Google Brain branch, together with other .com companies, are making forays into genomics computation. And so today we're going to talk about this recent announcement and the connotations that it has. To put a little bit of this in context, in the history of omics, together with compute, in the 1970s to 80s, we started accumulating with the scientific community a few DNA and protein sequences, amino acid sequences, to start studying at the molecular level these parts of the world, these parts of biology, DNA, RNA, and proteins. And some of the algorithms that are most well known of this pre-NGS era were developed in the 70s, 80s, 90s. Outstandingly, Smith-Waterman, Blast, Needleman-Bunch, etc. And so these developments that kept going through the 80s and 90s matched with the development in hardware and the CPU, both in the speed of the clock, but also in multi-core, multi-threading advancements, made the work that happened at that period of time, 20, 30 years, better and faster, because you could compute more with faster and more numerous cores in the computation that you were able to do. And then at the turn of the century, the millennia, we sequenced the human genome, the reference human genome. And a few years after that, the NGS era started, namely with the 454 and Solexa technology, the Solexa technology that ended up being acquired by Illumina, and it's the Illumina technology that we all know these days. So this happened, 30, 40 years after people started working hard on algorithms for bioinformatics before that. And then at that point, we had a lot more to choose from in terms of compute. GPUs and GPU acceleration had already started in the mid-90s. It's difficult to say where, but I would say in the mid-90s as a starting point, and became more pervasive later on. And so if you wanted to do any compute in the early 2000s, then you could either combine it with multi-threading applications with faster CPUs, but also with GPUs. And what has happened recently around the world of neural networks and AI is that companies have started being interested in accelerating that kind of application, neural network application, with these other accelerators that are either decoupled from the main CPU or coupled with it, or as a USB stick, for example. And then these accelerators that have the largest motivation in AI in companies like whatever Google trying to recognize images automatically, all the other .com and post.com companies, Facebook, etc., Amazon with voice recognition, they all are interested in doing that computation faster, and so these accelerators came to the fore because of that. But at the same time, they can be used for bioinformatic applications. And we're in the era now where, starting with the 1,000 Genomes project, we already have tens of thousands of genomes available to us, and then anything that speeds up the computation is valuable. And in this graph, we also saw the evolution of formats, file formats, and how those are important, and file format initiatives, how those are important to be able to generate data efficiently, but also at scale and with the right type of compute. And so in this tweet, Daniel Carroll says use methods that use AVX and will accelerate their applications, and then announces a deep variant version 0.7 release that shows improvements in speed due to this AVX instruction from Intel CPUs. And then Li Hang has also been working on this for his Minimap2 software, and he sees insignificant speedups over SSE, which is a technology that precedes AVX. And so he says, in terms of bandwidth, AVX 512 should be twice as fast as AVX and 4 times as fast as SSE, but he doesn't see that speed up. And then we're going to analyze later a blog post from someone else to see what could be the reason for that. So Andy Carroll says, yeah, interesting in deep variant. They use whichever acceleration is available. Better performance between AVX2 and Broadwell AVX 512, even more comparing to SSE. But TensorFlow likely disproportionately benefits due to the instruction profile, which is very important. So what is deep variant? Deep variant is something that was developed at the Google Brain, part of Google Genomics team at Google Brain, and it's an open source tool with the code available on GitHub for analyzing the DNA sequence of individuals and call variants. And it's built on top of TensorFlow. And so Andy Carroll, currently product lead at Genomics, Google AI, and previously he was for many years at DNA Nexus, VP of Science before he left, has been the one posting this. He's a Stanford Uni alumni. And then we're going to talk about two more people contributing to this conversation. Daniel Lemaire at Quebec, at Toulouse, Quebec, professor and department chair at the Toulouse of University of Quebec, and Li Hang, who is assistant professor of biomedical informatics at Harvard Medical School currently. He spent many years at the Broad Institute. He was a postdoctoral fellow and PhD student at the Wellcome Trust and BGI. I couldn't find a picture of him on LinkedIn, but here's a picture of him sat next to a much younger version of me, much hairier version of me as well, from a conference we organized and where he presented his software. And so deep variant, whatever version you're using, but the latest one, 0.7, what it does essentially is it analyzes the window around each candidate variant. For example, here, a GT change in this column by adding a 7 base pair window left and right, upstream and downstream of that variant or candidate variant. And then it represents these as using this alignment tensor. It encodes this alignment in a way that then can be given to an NN. And then there's an optimization function that basically tells you what's the probability of it not being a variant or being a het, in this case 0.98, or being a complex variant, etc. How does the variant perform versus other variant columns? Out of the list here, the variant is the one that was developed the latter. And in this plot, we show NovaSeq data at about 20x for a reference individual called G001. And here we show the errors for false negatives and false positives, both for SNPs and indels. And so lower the errors, the better. And we show here that, well, the deep variant team shows here that they have the lowest level of errors, both in SNPs and indels. And in indels, it makes quite a big difference, as you can see here. GATK4 being the second best in SNPs and indels. This is another one that shows the evolution of BIP variant from version 0.4, 5, and 6. And it's, in this case, for a PCR plus whole genome sequencing dataset done on a Hi-seq-X at 35x. This is important, PCR plus, because the data should be noisier. And this is something we've mentioned in other episodes. The best way of producing libraries for Illumina is to use the PCR-free library prep. But because if you do a PCR amplification in the PCR plus method, the polymerase introduces errors in the sample, and these errors are then picked up by the sequencer and shown to the next tool, the aligner and the variant caller. And so being able to reliably call variants on a PCR plus method, which is more convenient for many applications where you don't have enough DNA to do PCR free sample prep, then it shows an advantage. And the variant here shows better results than GATK and Strelka too. Strelka is the variant caller that was developed. It's still being developed at Illumina. When I worked for Illumina, I was part of the team, the Strelka team, evaluating it against other tools. At that time, mostly GATK. Does deep variant only work for Illumina data? No, it works for other data sets. And here there's a comparison showing Illumina NovaSeq data and the BGI SIG 500, which is from the BGI MGI company in China, who have developed this sequencer that looks very similar in chemistry and technology to the Illumina sequencing technology. And so here the question being, can deep variant do well here? And then for the blue samples, BGI, compared to the greenish samples, Illumina samples, yes, the variant does very well, better than Sentient, Strelka, GATK4, current version of GATK and Freebase. And the BGI technology is a little bit worse here in terms of total number of FPs, FNs, but very comparable to the Illumina technology. And here, for example, GATK and Freebase, sometimes they get it wrong more times with Illumina technology than they do with the BGI SIG technology. Obviously, the quality of these samples that says here Illumina NovaSeq operated by BGI can vary and can produce different results. And finally, the variant also works on PacBio CCS data. And here's another plot showing precision of recall and the F1, and it shows that at around 20x coverage, you've already hit a very good mark in terms of this ROC curve using deep variant. And at 25, 20, whatever, 7, 8x, you get very good scores from calling variants with deep variant. So back to the instruction set. This AVX 512 is an extension of the AVE SIMD instructions that were added to the x86 instruction set architecture by Intel starting in 2013, and then they implemented it, rolled it over into different iterations of their chips, the Xeon Phi, the Skylake, the Core X series, and the Xeon Scalable processor family, etc. And they've recently introduced it in both chips like the Larbi Phi co-processor, which is can be attached to a processor, but it can also be like a USB stick completely decoupled from the other hardware. And how does this compare to previous technology? So AVX12, 512, VL, DQ, BW, different subtypes of it. Present in Skylake 2017, was also present in Night's Landing 2016, but then the only thing that was present before that was the AVX2 or AVX or SSEs. And so people that write software have a lot more experience with SSEs and AVX than they have with AVX2 or AVX 512. And here's a graphical representation of what the different types are, which essentially show how much bigger the registers are, right, for storing these the data to perform the compute, the operations. And then so this release of Deep Varian shows a threefold improvement in end-to-end speed, and this they say is by enabling to take full advantage of the AVX 512 instruction set. And it shows how this new instruction set can accelerate and have an impact in applied research, in this case in genomics. They say that the size of the register is important for the acceleration, and that they can operate, because of the size of register and the way it's packed, they can operate on more information at once, right? And so they compare the speeds of the different versions of the variant they've been working on, and V07 is actually slower than V06 without optimizations, but then with optimizations, it is 3 to 5 times faster than the other versions, the same version, not optimized, and the previous version. And so an important point here, and this is a slide which I believe comes directly from Intel, is the frequency behavior of the chips containing the AVX code. And this is something that's going to be mentioned later on in the blog post by Lemaire. It says that the frequency of each code is determined independently based on the workload demand. And however many times you call the AVX instruction set, will have an effect on the frequency of the running code. And so they have different turbo frequency limits. So the idea of releasing the different incarnations of AVX was to speed up the computation of these floating point operations. And initially, they wanted to have a four times faster SSE with AVX 512, 2 times faster than AVX2. And part of this is going to be compilers, which we're going to talk about also a little bit later on. So where is AVX apart from in the Deep Varian code? It's also in GATK4. And this was a joint collaboration between the Center for Genomic Data Engineering and Intel, the company. And they showed an increased performance in a specific part of GATK4, part of haplotype caller. Haplotype caller allows you to call single samples, and it's very good at doing it. And so this pair HMM part of the code showed an optimized runtime based on making use of the AVX 512 instruction set. It's also in Minimap, or put it this way, Li Hang is trying it on Minimap, and in this PDF of the Minimap 2 software, Li Hang explains how he enabled the vectorization of certain compute by slightly modifying the code. And this was initially done on SSE because Minimap 2, I think, was published at least a couple of years ago, and parts of the code could have been worked earlier than that. So software that was developed and implemented to take advantage of the SSE instruction set. Now Li Hang is looking at AVX and AVX 512 to see if it increases performance. And what he sees is that he doesn't see this 2x, 4x improvement over SSE. And he saw a big leap on the SSE side of things. So Lemaire in his blog says that a lot of this is to do with frequency reductions, based specifically on which instructions you use and how many cores are active, downclocking. And then he says some engineers have recommended to disable the instruction set altogether to get full advantage of the servers, and he'll make a conclusion based on his analysis at the end to see if that's adequate or not. So he says that worrying about AVX 512 downclocking, one should worry about it as long as you operate on shorter 128 or 5 or 256-bit registers, because AVX512 can do more than that, and so you're not going to get full advantage of that. It says downclocking happens per core and for a short time after you have used a particular instruction, for example, about 2 milliseconds. And so if you just use light AVX512, even if it is across all cores, then the downclocking, automated downclocking, is modest of about 15%. And then he looked at also the software that has to be compiled, and the compilers in the software that has to be compiled with these new instruction sets. And then he says that LLVM Clang goes very aggressive in using this, whereas the ICC compiler from Intel and the GCC compiler from the GNU organization, you have to specify in a specific flags if you want to make use of the instruction set, right? Unless you put this flag, this parameter when you compile your code with GCC, then it's not going to change the way the code is compiled to try and make use of the new instruction set. So as a future, what it says that operating systems or application frameworks could assign threats to specific cores according to the type of instruction they're using, and sort of spread the load of how much of the AVX512 you're calling to ameliorate the downclocking. And he says it's a bit too much to unwarranted to disable AVX by default, and there's future work to be done here to make better use of this. And so Lemaire and the co-author Travis Downs here explain the way they see this performing. And so, yeah, this was the initial tweet that instigated me to talk about these optimizations from Andy O'Carroll and the conversation with Li Hang about what they see, the optimizations they see in their respective code bases. I hope you enjoyed this episode, and you can follow me on Twitter, twitter.com/Alberto, one word, name and surname. And please contact me through LinkedIn if you want. I'm happy to answer any questions you have. And a reminder that this slide deck is available upon request. And I hope you enjoyed this episode, and I hope to see you in the next episode of the Omics Cast.